Okay, we're going to chug right along. Now introducing Dr. Danielle Veenma, talking about eye tracking in children with grin disorders. Thank you.
So, good afternoon. So, thank you for the invitation to have a presentation. So, I will be speaking on behalf of the GRI Expertise Center at Erasmus MC, the Children's Hospital. And I will present part of our ongoing research in which we explore whether eye tracking as a possible outcome measure in children with GRI-related disorders. So, our primary focus is first on visual development and visual perception, especially children at risk for CVI, so for cerebral visual impairment. And this work is in collaboration with Dr. Johan Pell, who is a neuroscientist at our hospital at the Vestibular and Oculomotor Lab at Erasmus MC. Next slide. So I have no conflict of interest for this topic presented here. Next slide. I will briefly summarize the workflow of our expertise center. So in principle, each Dutch patient with a genetically confirmed diagnosis of GRI will be referred to our team at Erasmus. And then at that point, we will perform a multidisciplinary assessment on what we call key ages. So these are key ages during childhood. And then last year, we also created an adult Encore GRI clinic. So we now want to ensure lifelong follow-up for our natural history data collection. And next to this more clinically oriented assessment, all GRI patients are now asked to also enroll in our Brain Lab research program, which is marked here in yellow. Next slide. So the research goal for the Dutch Brain Lab is to identify outcome assessments that are clinically relevant, for example, to inform about prognosis or to guide clinical management, but additionally can be used to monitor treatment responses for future trials. And for this, we make use of the Dutch Brain Lab at our hospital, and it's a testing facility that consists of 3 rooms, and it's designed not to focus on a specific condition, but on assessing the level of functioning of our patients across different developmental domains. And we use like teams of all the hospital and also the technical university. So we try to invent and come up with new techniques, state-of-the-art techniques. And similar to our expert clinic, we enroll these patients and we view them and have multidisciplinary assessments again at these key ages. So it's our intention to repeat it and then, as kind of a longitudinal cohort follow-up design. So today I want to show you the results of our eye tracking assessment. Next slide. But before I show you the eye tracking protocol, I need to explain a few concepts. So first of all, visual processing, so how we perceive the world around us and interpret it. It's a very complex system. It involves many different brain areas that are depicted here on the left, and more schematically and simplified on the right. So this right-hand picture shows how different brain regions work together during a process of a visual stimulus. So first, the visual input arrives at the primary visual cortex and then at the back of our brain, and we call this our sensory entry point. And then from there, the information is relayed to the posterior parietal cortex, which kind of helps also direct visual attention. And from that, it's directed to the frontal eye fields, so the brain area where the eye movements are controlled. And where you plan your eye movements. So when we measure eye movements, we're not just testing the eyes, we are also tapping into a much broader network that involves visual processing, attention, and motor control. Next slide. So why eye tracking also? Well, this image on the left hand, it also shows you how the visual system develops over time. So from 20 weeks gestation into adulthood. And the orange area show you here in how many areas are actually involved in visual and visual processing. And you can see how these areas mature and also expand significantly from the 3rd trimester of pregnancy and then continue to develop after birth. So you can also see that like almost 40% of your brain supports visual functioning. So making it, as we view it, kind of a potential, like a sensitive early marker for overall brain development. What we also showed in earlier work and also from others, like eye tracking assessments are non-invasive, they are very fast, they are well tolerated, and you can do them from a very, very young age, even like, around 12 months of age. So we have other expert clinics. My colleagues at the Angelman cohort clinic, they also performed a feasibility study in Angelman syndrome's children that are nonverbal, and they showed that it is also really possible to do in these more severely impaired patients. And then the third reason to also include it, like we know CVI has been assessed already as a problem in both GRIN 1 but also maybe GRIN 2B patients. We wanted to explore how this would be at a group level. Next slide. So what is our eye tracking protocol? So what do we actually measure? So the basic idea is that you're gonna measure how quickly and how accurately a child can move its eyes in response to a certain visual stimulus. So by presenting the stimulus at different locations of the screen, or by using different backgrounds or contrast, you can test different items of the visual field itself, but also things like color vision. And so our basic eye tracking protocol consists of these different stimuli that you can show, that you can see on the left. So it's contrast, color, locomotion, fixation, and smooth pursuit. And yeah, the video was already running. So on the right side, you show it shows you a little bit what the children are also seeing. So they start with a salient picture like a kangaroo, and then you go to the next stimulus with color vision, and then fixation and a smiley. So we go repeatedly through these different cycles to grasp the basic items of visual processing. You have to keep in mind that so we can perform these tasks at a very young age because we do not depend on verbal skills, and a total protocol takes about 3 minutes in total. Next slide. So when we process these eye tracking results, what we do, we are going to calculate what we call the reaction time to fixation, so the RTF, and then we do that for each stimulus. So another way to put it is you look at how long each child, or how long it took for the child to fixate on a specific visual target after it appeared. And next to this RTF, we can also analyze the way, like the pattern, how they fixated. So whether the eyes moved in certain ways and expected ways. Next slide. So in typically developing children, the results of this like basic visual processing protocol, it looks like this. So on the right hand, so on the y-axis you have the RTF, on the x-axis you have the age, and each dot now represents this RTF of one child. And then the different colors show you the different visual stimuli. And what you see, it's kind of a structured and also layered picture that evolves. So it means that, you know, children can observe fast, and they have rather consistent fixation times for all these different visual stimuli. So it's efficient, and they can do that really, really quickly. Next slide. So what we then did, this shows results of research which was performed in the hospital at the neonatal unit, which we know premature born children are at risk for CVI. So I hope you can appreciate that showing again the same picture as I showed you before, that premature born children have slower fixation time, and you can also see that it's less accurate. They just have a reduced ability to follow visual patterns. So this study already showed us that eye tracking can help you to detect CVI earlier, especially in very young, as of a young age. Next slide. This from this first preliminary data, we compiled the RTF data for all stimuli. So you can kind of have a normal curve for all these different visual stimuli, which I show you here on the right. And I hope you can, we want to use this as our standard when we're going to measure our GRI children. So next slide. So we postulated that with this basic visual protocol, we would be feasible in a majority of the GRI and the KMK2 children. And this hypothesis was based on our Angelman cohort. And we defined feasibility that for each stimulus, the child would be able to see 25% of the stimuli, and then with what we call a minimum of 40% of the gaze data. And secondly, we suspected and hypothesized that GRIN children would have significantly delayed visual processing speed, and maybe that also the patterns would be distorted in these children that are impaired. Next slide. So these first results show the result for the feasibility. So the first enrollment period was between September 2023 and February of this year. The age range was between 6 months and 18 years. And the sex difference was the same. I do not have time to go in detail, but we measure also neuropsychological assessments. We do EEG. So in the future, we also want to correlate it to other measurements. And so for our first hypothesis, well, is it even feasible to try to do this in this population that is easily distracted and can be, behaviorally challenging? And the answer was yes. Like in the majority of cases, we could get good quality data, and it was not depending on their motor skills or their verbal abilities. So in the next slide, I want to show you the initial RTF results for one of the stimulus, which is the cartoon stimulus. Again, on the y-axis, you have the RTF, and on the x-axis, the age of the children, and then again, each dot is one child. And what you can see, so a majority of the cohort has normal RTF times, but there are children that show these slower RTF times. So it shows that this subcohort of children is at risk of CVI or have CVI. And so this also highlights that it not can only be used for cohort and for group level of studies, but we can also use it for in the clinic itself to help maybe get more personalized suggestions for these children how to improve their skills. So next slide. So this is the compiled data for all the stimuli. So again, on the right corner, I copied the picture for the normal children. And I hope you can appreciate so that some of the layering, what we are seeing with the normal children, is also happening in the GRI and the KMK2 children. But again, it's not for subgroups are at risk. So we need to really see how this for the larger, we need to collect more data, more children. So next slide. So why does this matter? So we believe that this eye tracking can offer us like a functional and also early quantitative marker for visual processing. We believe that the eye tracking protocol so holds value for both the children on an individual level, but also for cohort research for GRI. And especially eye tracking can help so detect CVI for children at a very young age. So what we want to do next is, so this is just a pilot study, and we would like to try to expand our data set. If you are interested in, and maybe you can go to your ophthalmology department and ask, because many of them sometimes also have these eye tracking machines. So we would be happy to share also our protocol, and we're working on an automated analysis pipeline for this. And also in parallel, we are measuring this with the children also having a 126/8 channel EEG on it. So we are also, what I said before, like trying to look at it from results from multimodal assessments that we do. So next slide. The next slide, I already talked about this. So now what we want, we're going to follow up the children in a different time points. So eventually we want to develop developmental curves for all the children. And the next slide. And we are also now working on a protocol so this is the basic visual processing protocol that we have, but now add also performance task. And sorry, the next slide. So one of the tasks that we are thinking about is, for example, emotion recognition, that you give different pictures to the children and then see whether they can recognize certain emotions or not by looking at how they are attracted their attention. And next slide, in even higher functioning children, you can think of different kind of sequence learning tasks that you can also use the eye tracker to see how they perform and see whether these can deliver outcome measurements. So next slides, and the next one, sorry. So again, this is, I hope to expand the work, and if any of you is interested, please email us and we can hopefully collaborate. Thank you.
Okay, so our next two speakers are actually on Zoom. Thank you guys so much for joining us. We really appreciate it and bearing with our time differences here. So we're going to start with Moriam talking about understanding GRIN 2D from patients to a mouse model of hope.
At 3 weeks of age, the motor deficits become more pronounced. This is also the age when pups start to eat solid food and gain independence. And at this stage, mutant mice show strong and persistent clasping response, which continues into adulthood. In another motor test, the rotarod, mutant mice fell off the rotating rod earlier than wild types, indicating deficits in coordination and muscle strength. And these differences remain consistent as the mice grow older as well. We wanted to test other aspects of motor coordination, and in the bulk catalepsy test, mutant mice again show clear deficits. Catalepsy reflects an inability to correct abnormal posture and is often used to assess motor rigidity or impaired sensory motor processing. In the open field test at 4 weeks of age, we also saw clear signs of hyperactivity in the mutant mice. They showed increased locomotion and spent more time moving than wild-type controls. The cognitive symptoms appeared after the motor deficits, emerging slightly later in development. And the first sign we observed was increased anxiety. At 4 weeks of age in the open field test, mutant mice spent significantly less time in the center of the arena. And this avoidant behavior is commonly interrupted as anxiety-like and marked at the beginning of the cognitive and emotional phenotype in our GRIN 2D mouse model. At 5 weeks of age, just as the mice begin sexual maturation, we observed additional cognitive symptoms. In the Y-maze test, which evaluates spatial and working memory, mutant mice show clear impairment compared to wild type. And this continues the progression from early anxiety-like behavior into broader cognitive dysfunction. So to summarize the behavioral findings, we saw a strong overlap between the symptoms in our mouse model and those reported in patients, including seizures, motor, and cognitive deficits. And this close match makes the green 2D mouse model a very powerful model for studying green 2D related developmental epilepsies. So now that we've covered the behavior, let's move on to electrophysiology. We perform patch clamp recordings from Purkinje neurons in acute cerebellar slices. And patch clamp is a technique that allows us to measure the electrical activity of individual neurons with high precision, giving us direct insight into how they fire and respond to stimuli. And we focused on the cerebellum because of the clear and early motor symptoms, and because this brain region plays a central role in movement coordination and motor learning. Within the cerebellum, Purkinje cells are the principal output neurons, and they integrate information from across the cerebellar cortex and control downstream motor pathway. So studying their activity gives us a window into how motor circuits may be disrupted in green 2D. The first thing we observed was that at 2 weeks of age, Purkinje neurons in the mutant mice had a lower spontaneous firing frequency compared to wild types. And this reduction in activity aligns with the early motor deficits we saw behaviorally and suggests that the cerebellar dysfunctions begin early in development. Later in development, the firing frequency returned to levels similar to wild types, which may indicate the emergence of compensatory mechanism. However, by that point, the motor symptoms were already well established, which suggests that early disruption in Purkinje activity may have lasting effects. We also looked at how the cell responds to NMDA. And while there was no differences in young mice, older mutants show a stronger response to NMDA compared to wild types. And this aligns with previous report that this green 2D mutation causes a gain of function effect. And here we see it reflected in neuronal activity. Next, we looked at the morphology of these Purkinje neurons, since changes in structures can often reflect or contribute to functional abnormalities. And we found that mutant mice had smaller cell bodies compared to wild types. And this suggests that although the overall wiring of the cells may be preserved, there are still subtle cellular changes that could affect how these neurons process and transmit information. Altogether, the electrophysiology data show that the green 2D mice have early firing deficits and a bigger response to NMDA later in life, possibly helping to explain some of the behavioral findings. Unlike patch clamp, which we usually don't do on humans, EEG is a widely used clinical tool. So we recorded the brain activity of our mice with EEG and hoped it would offer some more translational insight. And green 2D mice showed near constant epileptic spikes, with abnormal background activity that was present almost all of the time. Unlike other models of DEEs, where seizures occur as a distinct event, our green 2D mice displayed ongoing epileptiform activity in the baseline signal. So to better understand the nature of this ongoing epileptic activity, we moved from observing the EEG signal over time to analyzing its frequency components. This means we use the mathematical tool to break the complex EEG waveform into its underlying frequency bands, delta, theta, alpha, beta, and gamma. And by looking at the signal in the frequency domain, we could detect subtle and continuous abnormalities that aren't always obvious in the raw EEG trace. So we performed this frequency analysis and found reduced power in the delta and gamma bands and increased power in the alpha, beta, and theta bands, starting as early as 4 weeks of age. And we wanted to examine whether the spectral features we identified are relevant to the human disease. So we analyzed human patient EEG using the same methods and found similar abnormal frequency patterns in both human and mouse recording. And this overlap not only shows that our model is a good fit, but also suggests that these EEG abnormalities are specific markers for green 2D developmental epilepsy and could be used to track the disease and evaluate treatment. So given the clear and disease-specific EEG signature we identified, we next set to assess the effect of potential treatment. We tested 3 FDA-approved drugs, ketamine, memantine, and phenytoin, all of which either modulate NMDA receptor activity or are widely used in the treatment of epilepsy. We began with ketamine, a non-competitive NMDA receptor antagonist, and although it's used clinically for certain seizure types and as an anesthetic, in our model it had a very clear negative effect. It triggered seizures in all of our mutant mice and showed no improvement in the EEG activity. Next, we tried memantine, also an NMDA antagonist, And here we observed an increase in the delta power, suggesting partial improvement in the background brain activity. We also tried phenytoin, classic anti-seizure medication, and phenytoin produced a similar partial rescue in the delta band, supporting its potential benefit even without targeting the mutation directly. To better understand the memantin's effect, we conducted some electrophysiology cortical recordings, and Arno memantine, the epileptic spike, were shorter in duration, and the overall amount of epileptiform activity was reduced, pointing to a measurable, although incomplete, therapeutic effect. So while none of these specific treatments led to a full rescue of the phenotype, they show that our model and tools that we've developed can be used to evaluate the treatment efficacy in quantifiable ways. And this opens the door to more precise mechanism-based drug screening and ultimately and hopefully to the development of more effective therapies for GRI2D DEEs. So to summarize, our mouse model captures many of the core features of GRI2D developmental epilepsies, including spontaneous seizures, early mortality, motor impairment, cognitive symptoms, and abnormal brain oscillations. Importantly, the symptoms follow a clear developmental trajectory, starting with motor deficits, hyperactivity, and then progressing to cognitive impairments. We also identified EEG markers that could be used to evaluate future treatments. Our paper has been published in Brain, alongside the complementary study by Teo et al. From Wayne's Franklin Lab at Columbia University, which focuses on hippocampal dysfunction. I want to take this opportunity to thank them. The Franklin Lab are the ones that developed this mouse model, and we're very grateful for their collaboration. Together, the two papers offer a broader perspective on how GRI2D mutation affect brain function. Much of what I share today is described in full details in the paper, and I highly recommend reading the Frankel paper as well. I'd like to thank my supervisor and my lab mates. Many of them contributed to the work I share today. I also want to thank the Green Foundation for their ongoing support and for bringing together families, clinicians, and researchers around the shared goal. And for me, this work at its core is about people, about life that can be changed through science, through collaboration, and through hope. Thank you all for your attention and for the opportunity to present here today.
Thank you so much. We're now going to have our last speaker of this session, Danielle Galber, who's going to be talking about investigating therapeutic avenues for GRI2D linked developmental and epileptic encephalopathies.
So firstly, thank you for having us. Obviously, I would much rather be there instead of staring into a screen, but I'll settle for this. So my name's Danielle, and I'm doing my PhD with Moore at Tel Aviv University. And what's really nice is I don't have to do an introduction because Moore introduced our work. So I'll just say that I'm also working on the valine to isoleucine mutation, and we're working on treating these beautiful children. So the work I'm going to present today is split into two sections that are both focused on therapeutic avenues that we are going down. So the first thing I'm going to present is molecular dynamics that we are performing in order to understand the structural conformation of the receptors and understand the structural effects that this mutation has on receptor functionality. And then I will talk about a drug that we are currently in the process of analyzing. So let's begin by talking about molecular dynamics. So in order to perform molecular dynamics, we needed a model. And at the time, the full structure was unavailable. So we created our own model based on a rat model of NMDA receptors with gluN2B. And obviously, as science goes, once we'd finished this, the full structure was actually solved. So we compared our model with the full structure, and we found that they have a very low RMSD between them. So we continued working on our model. And we ran 3 simulations of the wild type and the mutant receptor, as can be seen here. And we performed various analysis in order to see if there was any differences between them. And one of the most interesting things we found was this potential divergence between the two receptors. So I'm not sure how well you can see it, but don't worry, I'll go into more a better quality in a second. But we can see in the circle two amino acids of GLUN1 and GLUN2D that in the wild type seem to have no interaction between them, and in the mutant receptor do have some sort of interaction. So we wanted to understand what this interaction was, and we found that there is a salt bridge that is formed between a lysine in GLUN2D and a glutamate in GLUN1. And in order to see exactly what was going on, we basically took our simulations and broke them down into 1,000 frames and calculated in how many frames we observed this interaction. And what we can see in the top section is that this interaction barely exists in the wild type. But then when we look at the mutant underneath, we can see that in at least one of the pairs, as we know, it's a tetrameric receptor, so we have two sets of GLUN1, GLUN2D pairs. In at least one of them, in the mutant receptors, this salt bridge seems to exist. And in our second replica, this actually exists in both of the pairs. So what's really interesting is this salt bridge, as I said, is from a lysine, and that lysine is placed at 656, which is only 11 amino acids upstream from where our mutation is, indicating that this could be a good candidate to help us understand the structure, the effect on the structure that this mutant has. So what we've done is we've identified it in silico, and now we're working on validating it in vitro. So we created an expression vehicle and introduced the valine to isoleucine mutation, and then also introduced another mutation in this lysine in order to break the salt bridge. And we're currently in the process of expressing it in oocytes and performing 2 electrode voltage clamping in order to measure the receptor activity. So now jumping to the other part of my presentation, we are in the process of analyzing a drug called Compound A. And this drug was found to reduce premature death and the seizure burden in both Dravet and LGS models, which are similar DEEs. So what is this drug? Compound A works on the pathway that converts brain cholesterol to 24-SHC, which is done by an enzyme called cholesterol 24-hydroxylase. And compound A actually inhibits this conversion, reducing the levels of 24-SHC. So in addition to the positive therapeutic effects that it has in similar disorders, 24-SHC is actually a positive modulator of the NMDA receptor. So we hypothesize that if we lower the levels of 24-SHC, we'll also be able to lower the levels of this hyperactive activity of the receptor. So what do we do? Our goal is to treat the mouse model that Moore presented just before me and do a general analysis. So we wanted to measure the levels of 24-SHC in the brain, analyze survival rates, measure the effect on cognitive and motoric performance, evaluate the seizure burden, and then overall work out if we can understand the mechanism behind this therapeutic activity. So what we did was we treated our mice via chow. So we added the drug into the mouse food. And we had 2 different age groups. We had mice, P0 mice that were treated from birth, who were born to parents who also received the drug. And then we have mouse that were mice that were treated at weaning age, which is slightly more clinically relevant, who received the drug from a P21. So to begin, what we can see here is that compound A does indeed reduce 24-SHC levels, indicating that the pathway that I presented previously does indeed work. The compound A passes the blood-brain barrier and reduces 24-SHC levels. So the next thing we wanted to do was measure if there was an effect on survival. And here, excitingly, we can see that yes, compound A does protect against premature mortality. If we look at the green part of the graph in both P0 mice and P21, we can see that 50% of our mouse model do not survive past 40 days, whereas mice that are treated in both age groups 70% of them actually reached the endpoint of our experiment, which we chose to be 150 days. So there's a significantly rescued survival rate here. The next thing we want to do was measure cognitive and motoric impairments. So here we can see sample experiment where we measured, we performed a rotarod, which measures the mouse's ability to remain on a rod that is rotating at accelerating speeds. And as more presented before, we can see that our mouse model exhibits a significantly impaired balance. And our compound A seems to rescue this impairment, with the rescue being stronger at P0 in the P0 mice. However, there seems to be a trend in P21 too. Another thing we performed was measuring hyperactivity. Once again, more presented that just before me. Here we measured the mouse's locomotive activity in the open field arena, and we see that these mice, our mouse model moves a lot further than the wild type siblings. However, this is rescued with treatment. Again, the stronger effect is in the younger treatment group. So the next thing we wanted to do was check the EG. Now, these results probably look very similar to you guys because more actually presented these exact results. So just as a recap, what we see is that in our mouse model, there is, when we take down the EG and we break it into the different components, the delta band is decreased and the theta band is increased. So we wanted to see if this was something that the drug could affect. And what I'm showing now is data that we're still collecting. However, I did want to show it because we can see that there seems to be some sort of improvement with at least the delta being increased. So finally, we wanted to understand the mechanism behind this therapeutic activity that we seem to have found. And we decided to work on the inflammation pathway because in various other similar disorders, there is increased inflammation. So we wanted to check the inflammation profile of our mice. And in order to do this, we stained against GFAP, which is a marker of astrocyte activation. And what we can see here in green is that our mouse model exhibits a significantly higher astrocyte activation. So then when we took brains from our treated mice, we actually see that this astrocyte activation is reduced, indicating that this could be a pathway through which the drug works. So just to summarize, we have compound A. We found that compound A protects against premature mortality. It rescues motor and cognitive deficits. There seems to be a partial correction of the abnormal brain oscillations. And we've discovered that there is a reduction in astrocyte activation, which could potentially be the pathway. So all in all, we've actually found a drug that has a novel focus on the behavioral phenotype, because as most of you probably know, currently in the market, the drugs that are used to treat patients focus solely on the epileptic phenotype, and the developmental and behavioral is kind of left on the side. So we found something that focuses on both the phenotypes and has an effect in various intervention levels, with a stronger effect being in early intervention. So just to summarize everything that I said in the first part of my talk, I told you guys about a potential interaction that we found between GLUN1 and GLUN2D that could help us understand the structural effects that this valine to isoleucine mutation has on our receptor, which one helps us understand the mechanism behind this pathology, but it also opens up an additional target that we can use to treat this disorder. And secondly, I've presented compound A, which is a novel drug that we found that has the potential to be a really interesting target to help treat patients and improve their quality of life. And so thank you so much for listening. I want to just take this moment to say thank you to my two amazing labs. I'm really lucky to have both Karen and Professor Karen Abraham and Professor Moran Rubinstein. And thank you all again for listening. And thank you for having me.
I think you guys can hear us on Zoom still, so I'm going to open it up to questions and bring Danielle Veenma back up here. And we're going to give this a go and see how it goes with Zoom questions. Thank you for bearing with us on technology issues. You got it? Okay, perfect. Any questions? Okay.
This is a question for Danielle. Are you at the liberty to telling us what compound A is?
Yeah, sorry, I wasn't sure which Danielle you were talking about. So it's a drug that we are working with Takeda. We're not at liberty to say exactly what the drug is, but as I said, it's an inhibitor of cholesterol 24-ester hydrocholesterol.
Okay. And I guess the other question that I had is, you think this could also be a possible therapy for other gain-of-function mutations in other NMDA receptor subunits?
So ideally, yes. I think the fact that it was found to be have therapeutic potential for non-NMDA receptor disorders means that the chances of it working on similar disorders to what we found is quite high. That's definitely something that we want to look into.
Thank you.
I may have missed it, but at the end of the last talk where you jumped to astrocyte activation, I was wondering why the focus on that. Was that the most obvious phenotype, or, yeah, that's it.
Great question. It was kind of one of those situations where we had heard that in similar disorders there is increased inflammation, and we had an antibody for astrocytes. And also part of the work Takeda did, they found that the drug that we have could potentially work on inflammation. So it kind of came hand in hand, and we had our mouse slices, and we were lucky, I guess.
Yeah, okay.
Thank you. I've got a question for the other Danielle about that task that you've got for the eye tracking. Is there a possibility of extending that sort of task to make it more of a learning task? I'm just thinking that there wasn't too much of an effect compared to sort of your curve, and I'm just wondering if there would be more of an effect if you could extend it to.
It was my first reaction, and with I think the neuroscientists, what I'm working at. So the data I showed you is really like investigating the basic functioning of this visual system, right? So so it's not like it is measuring any of the cognitive or functioning of these children. And I think the difficulty, so there are already some tasks in the protocol that we're trying, but because you have so many different factors, like whether the children have their attention to the task, right? And you have verbal and non-verbal children, so we're now thinking about adding maybe some that they can point to things. So part of the Greek cohort will not be able to do that, but part will, so that we can like maybe add a few. So we're adapting this protocol to see, for higher functioning children, the sequence learning task will be possible, I can already tell you. But for the lower functioning group, we're thinking about the way to do it. And this is just, adapt and learn. But I am confident that we will find some of these tasks that can measure these higher performing things. And I had the same reaction, but like the specialist also said, well, I think it's important because, you know, when you think on how to help these children, well, if you don't process visual stimuli right, like you're gonna fall or you have issues with balance and things, right? But now we learn that at least subcohorts of GRI don't have those, so that means we can, the way how we learn them and show them things can be through visual stimuli. But there are in the GRIN2B, I think like 70% of the GRIN2B had faults in this basic visual processing. So I think I learned from the first literature, GRIN 1 was CVI, but now for me, we have to look at the GRIN 2B cohort as well. Yeah.
Thank you.
Yeah, so I'm also following up on the eye tracking. So it's super cool. But I also just wanted to follow up on some, questions on how to approach, what is the difference? How do you get out behavior versus visual, factors, call it like this?
Yes.
And then also, did you do some work on taking the same child multiple times within a short time interval to see how stable the signal is? And then also, it looked to me a little bit more qualitatively how the deviation from these curves was interpreted, and how to develop a statistical test which shows the deviation in that space in a way that it can be used for an endpoint in the trial, right? Because there's also people age, right? So they're exposed to stuff and, thoughts in this space. And then for the GRIN 2D crew, I would be interested to see if the team looked if the variant was tested in other subunits, because, function is often very preserved across NMDA receptor subunits. And one can learn maybe from GRIN 2B or GRIN 2A if the same residue has been tested.
Does online want to answer first? I'm fine. Well, I will. So first, for the question, I think you're totally correct. I think part maybe some of the issues with these children will be attention problems or something. But what I showed, in the visual protocol, there is already repetition in these tasks. I didn't have enough time, but how we do it, it's showing these tasks multiple different times in iteration. So that has been taken account for. And also from the normal standardized curves, I think, when you're really deviate from these ones, you have slower visual processing. That has been already tested in different cohorts, so I'm sure about this data. I think that the next big question is that we have to do the follow-up, right? We have to see these children in multiple, over time, and that is within the protocol, so we're going to do that. So I think part of it will probably resolve over time by itself for the basic visual processing, and therefore these more complex tasks come into the picture. And I think there is going to be the true value of maybe this as an outcome measurement. But we first had to prove that if your visual processing doesn't work correctly, you can't even think about a task like this, right? So we're now working with a team to come up with more complex and to be honest, on your question on behavior, to me, it is also a little bit the natural history. This comes back, I think, to the feasibility criterion. If we only develop tests that the children can only do in the most perfect circumstances, well, that's not a good protocol, I think. So I think you have to take into account that for some of the outcome measurements, subjects will fail, but so the tasks should be, so we have this feasibility criterion that at least 60% or 70% of the cohort should give with a certain task a good result. And I think that's how we approach it.
Yeah. I mean failing is also a phenotype, right? So we do a lot, failing is also a phenotype.
Yeah,
so
exactly.
We do tons of things with wearable, where we have the wearables with children with neurodevelopmental disorders, and we have similar issues like this.
Yeah.
But we definitely would love to replicate this.
Yes. Yeah. So email me.
and the GRIN 2D group?
So could you just repeat the question, because my internet obviously failed as you said it, so I was trying to work it out from Danielle's answer.
Yeah, of course.
But if you could just repeat it, that'd be great.
So most of the disease-causing variants in the NMDA receptor subunits, they are conserved and conserved residues. If you perform an alignment, then you see that it's the same residue. And often they have been tested in other residues for functional testing. For example, there have been more than 400 variants already tested across all kinds of NMDA receptor subunits. Have you looked if the residue you are interested in, which you modeled, has been molecularly tested in the other subunits, and what was the outcome of that?
More, do you want to answer or should I? Okay, it's not something that I can say 100%, like give you an answer. I definitely, it's something that we looked at in the past. And when we looked at it, we didn't see anything that was exactly the same. There are variants that are very close. It's in the TMD part of the receptor, and when you compare it to the others, there are very similar ones, but we didn't, from what I recall, we didn't see anything that that was an exact fit.
Great. Thank you guys so much for joining us via Zoom. We're wishing you well. Thank you, Danielle. Thank